Hello, biology, and welcome back to chapter 6. This one's all about photosynthesis. So, in chapter 4, we talked about the importance of different organelles in plants, and in chapter 5, we talked about cellular transport and homeostasis. We're going to take those two and we're going to mesh them together for your first kind of big chemical cycle. And this is a biochemical cycle where we're using both life and chemistry to make things. In this case, we're going to take sunlight and carbon dioxide and water, and this is the perfect recipe for plants to produce oxygen and sugar. There's a lot of steps on how we get there. This can be confusing. Take your time, watch it as many times as you need. Let's jump right in. So we need to talk about how organisms obtain energy, because we've talked about things use metabolism and things use homeostasis. Both of those things require the input of energy. So we can classify organisms by how they acquire energy. And the first classification is an autotroph. And autotrophs are things that create their own energy. They make it themselves. These are things like plants and algae. Other examples are in this picture here, like phytoplankton. And even some bacteria and protists are able to do photosynthesis as well. Now, there are two types of autotrophs. And the first autotroph we talked about was, the first one we're going to talk about, is chemotrophs. And these use energy stored in organic bonds from inorganic substances, from chemical bonds and inorganic substances. This deep sea thermal vent here is a great example. There's a whole ecosystem here built on these bacteria that use these inorganic sulfur molecules to create energy. And some scientists think that this is where the first life on Earth originated, is around these hydrothermal vents. The other kind is the phototrophs, and they are able to use light to create energy. Primarily, these will be from the sun. Humans in their fluorescent lives didn't evolve for several billion years, so they had to make do with what they had. But they're going to take this light energy and they're going to make it into organic compounds, primarily in the form of carbon, primarily in form of carbohydrates. The next class of organisms and how they get their energy are the heterotrophs, and you are a heterotroph. Heterotrophs get their energy from their food. So they eat plants, they eat other animals, and then convert organic compounds into energy. So even things that are herbivores, like ants, or other things like rabbits, are still heterotrophs because they're eating other organisms, even if they're just plants. So because of this, 98% of all the energy on the Earth comes directly or indirectly from solar energy. That other 2% is coming from those geothermal vents deep in the ocean. So 98% of it we're getting from the sun. That's a lot of energy, very reliant on the sun. Even if you aren't eating sunlight, you are eating things that have eaten sunlight. All the plants that you eat have eaten sunlight, and all of the organisms that you come in have consumed sunlight in some way or form. All right, so we're going to talk about photosynthesis, and this is what photosynthesis looks like as kind of like if you're a biochemistry student, this is what you're going to look like. But it's a complex series of reactions, and it takes reactants and turns them into products. We call it a biochemical pathway, meaning it's a life chemistry pathway, which is a series of chemical reactions where the product of one reaction is consumed in the next. So when we take Rubisco here, it turns into these things, which then turn into glycine, turn into serine, turn into glycerate, and continue around and around here. We're using, things are coming in, reacting, things are moving away, reactants are being pulled off, we're getting new products. We're going to go through it, we're going to talk about it, we're going to go through photosynthesis. So photosynthesis and versus cellular respiration. As a heterotroph, you get your energy from cellular respiration, and even plants use cellular respiration as well as photosynthesis. So phototrophs use carbon dioxide, water, and light to form glucose and oxygen. In the kind of opposite molecular level, we, as animals, use cellular respiration, where we use glucose and oxygen to make carbon dioxide and water and energy. So it's these kind of opposite things going on here. Now, cellular respiration is important because it allows plant and animal cells to use the energy that is stored inside of sugars, inside of carbohydrates. And this will be done in the mitochondria. The mitochondria is where cellular respiration is going to happen, and it's what we're going to talk about next chapter. All right, for photosynthesis, there are two basic stages to photosynthesis. The first is the light reactions, where we take light energy from the sun and convert them to chemical energy in the form of ATP, this little molecule that we've talked about before. It's kind of tucked up in here. But we'll make ATP, and we'll make the energy carrier NADPH. And NADPH is an energy carrier. It's a molecule. I'm not going to make you memorize its name. Just think of it as kind of this electron shuttle. The second stage of photosynthesis is the Calvin cycle, where organic compounds are formed using this carbon dioxide and the chemical energy that was stored in ATP and NADPH, this ATP and NADPH coming from the light reactions. We'll go that way, actually. Arrow will go around and down to the Calvin cycle. So something you'll need to know, this is the equation for photosynthesis, where we take 6 carbon dioxide molecules, add them to 6 H2O molecules, and we insert light energy to create this molecule, which this is glucose. C6H12O6. You need to know that. Glucose is C6H12O6. And plants also release 6 molecules of oxygen. This whole equation is important and may show up on one of your quizzes, so just a heads up. All right, capturing light energy. Plants have to first capture this energy before they can do stuff with it. So in the light reaction, this first stage of photosynthesis, it's named that because it requires light to work. Plants can't do this at dark. They need light to do this kind of reaction. The chloroplast, shown here, is an organelle that absorbs light. And they have 2 membranes. Chloroplasts have a bilayer, just like a cell plasma membrane, just like a mitochondria does. All right, next let's talk about the inside of the chloroplast. Inside of the chloroplast are these thylakoids, which are these flattened membrane stacks. Like this whole stack here is a thylakoid. Thylakoid, thylakoid. One of those is a thylakoid. They're arranged in these stacks that are called grana, which are connected, stacked thylakoids. And around them is the stroma, which is a solution or water that kind of nourishes and takes away the products of this reaction. Now, light from the sun is white, but it is actually a combination of all the light in the visible spectrum. So when light strikes an object, its colors can be reflected or absorbed by that object. Now, if you're seeing whatever color you see, like this curtain behind me is reflecting green light. It is absorbing all the other colors, and that's kind of the exact same thing that's going on in plants. These chlorophylls are absorbing all the blue and all the red, and all that's left over is this green in the middle here. So pigments is any compound that absorbs light, and it usually absorbs certain colors more than others. So it'll absorb all of one color and reflect the rest of it. And as you can see here, chlorophyll A and chlorophyll B have a little bit different reflection. So if you see a darker plant, it has more chlorophyll A, and if you see a lighter plant, it has more chlorophyll B. The chlorophylls are the most important of the pigments that plants use. There are other pigments called accessory pigments. We'll talk about them briefly. But there are 2 common types of chlorophylls. There is chlorophyll A and chlorophyll B. Chlorophyll A is more responsible of absorbing the blue light, and chlorophyll B is more responsible of the kind of redder, kind of more vibrant lights. So chlorophyll A, like we said, more red light. Chlorophyll B, think blue, more blue light. Both of them put green back out in reflection. And this is why leaves look green, is because these chlorophylls are absorbing all the other colors and reflecting green back. So light reactions need chlorophyll. Chlorophyll is directly linked in these. It is the one that is capturing the light. Chlorophyll B, or the blue one, remember, captures light energy and gives it to chlorophyll A. Because of this, chlorophyll B is considered an accessory pigment. And as you can see on this chart here, these carotenoids are going to give things to chlorophyll B, which give them to chlorophyll A. And the carotenoids are things like, if you look at carrots, that really, really orange color that they have is a carotenoid. So this carotenoid gives it to the chloroform B, which gives it to chloroform A, which makes the actual energy. Speaking of carotenoids, these are our yellow, orange, and brown pigments. They also usually have an accessory function, and they capture light that chlorophylls cannot. As your chlorophylls A and B start to break down in the fall because of a lot less light, you start to see these really, really vibrant carotenoids in leaves. And that's what gives us our fall colors, our oranges and our yellows and all of these ones. They're always there, but most of the time the chlorophylls are absorbing more light, and so they're reflecting back that green. These ones don't absorb red. They're reflecting this red back out so that you can see it. So how do we take this light energy and make it into chemical energy? So once we capture the light energy, we convert it to chemical energy. And we store it in this energy molecule ATP and this energy carrier NADPH. And to do this, we have a kind of work between these chlorophyll and these proteins to store this energy. So what happens is we have these photosystems. And these are these cluster of pigment molecules and other proteins that kind of shuttle light and electrons around. We have the photosystem 1 and the photosystem 2. They contain the same kind of pigments, but they perform different roles to each other. This right here is an example photosystem, this whole kind of diagram here. All right, so let's talk about the light reactions and how it actually starts. So it begins when the accessory molecules in the photosystem absorb light in photosystem 2. Photosystem 2 is this one over here. Photosystem 2 was discovered second. That's why it's photosystem 2. Even though it's kind of, we read left to right, photosystem 2 is the one on the left. Inside of this photosystem, the energy is passed to chlorophyll A. Now, this light energy is forcing these electrons to enter a higher energy state. They're now excited, and they will leave this photosystem 2 and go down here to photosystem 1. We need to talk about real quick reduction and oxidation review. Remember that oxidation reactions involve losing electrons, and reduction involves gaining electrons. And these things always happen together. So when this one loses an electron, it becomes more positive. This one gains an electron and becomes more negative. All right, so the chlorophyll A loses electrons. So it is being oxidized. Some substance has to be reduced with those electrons. And that one is the primary electron acceptor. And this primary electron acceptor here is taking these and shuttling them along. And they are found in the thylakoid membrane. So the primary electron acceptor takes this guy here, this electron, and shuttles it down through this, what we call the electron transport chain. And this transfers from one molecule to the next. You'll see there's 3 molecules on the ETC here. What this is doing is each time this is transferred, this electron is losing energy. This energy isn't lost, it's transferred, and you're moving hydrogens from the outside of the cell to the inside of the cell. You're pumping these hydrogens in. Those positive hydrogens are attracted to the highly negative electrons, and that's where this energy is going. So, at the same time as this one's getting light over here, photosystem 1 is also receiving light. And this electrons will move up to an acceptor inside of that photosystem, and then they will start to travel down an electron transport chain as well. Where do they get new electrons from? Well, the new electrons are coming from photosystem 2. As photosystem 2 sends this new electron down here, it gets excited again and starts to go up and through again. And this process repeats on and on. So we need to talk about the primary electron acceptor in this one now donates this electron to this second electron transport chain here. And this one's on the stroma side, facing the inside of the thylakoid. Excuse me, the inside of the chloroplast. And it's combining its H plus with this NADPH to form the molecule NADPH. And that's happening right here in this molecule. This molecule is being reduced. It is getting that extra hydrogen added to it, getting an extra electron on it, becoming reduced. Now, we need to talk about, before we go on to this part here, which is the kind of gear looking part, we need to talk about where do we get electrons originally from this? Because we said that photosystem 2 gives its electrons through the electron transport chain to photosystem 1. But where do these electrons come from originally? So the question, what replaces electrons? The answer is water. H2O replaces these electrons. So we have this cool molecule here called the water splitting enzyme, and it splits water molecules into three things. It splits them into protons, this 4H plus here, splits them into electrons, these 4E minus, and it splits them into this molecular oxygen that plants breathe out. So with this, we're getting two really cool things. We're getting more protons on the inside. We've been pumping protons from the outside with the electron transport chain, and now we're getting more protons on the inside, and we're getting more electrons to continue to run that photosystem system to get more and more protons on the inside. And we'll talk about why that's important next. All right, so now we need to make this ATP. So to make ATP, we go through a process called chemiosmosis, where we're synthesizing ATP using chemical reactions. It relies on a concentration gradient of protons, and protons are those H pluses. And this whole time, we've been pumping H pluses into the cell, into the thylakoid, into the chloroplast there, and we've been using the water splitting enzyme to add 4 more H pluses every time that we split a water. So what's happening is we're getting lots and lots of hydrogen on the inside of this thing here. So since this concentration is higher, we now have potential energy where these hydrogens want to go out of the cell. This molecule ATP synthase here will take these hydrogens, it will send them through this kind of pump here, and what happens is then these ADPs is kind of meshed together with a phosphate to become ATP. So we're using this hydrogen gradient to create energy. So what we're doing is we're taking this ADP plus an inorganic phosphate, which you'll sometimes see wrote PI, and then plus a hydrogen from the inside of the thylakoid here to make ATP. This energy is created from these protons moving across the thylakoid membrane against with their concentration gradient. And it's facilitated by ATP synthase. Now this is the end of 6.1, but remember that we've already created this NADPH and this ATP, and it's going to come back when we talk about the Calvin cycle next. All right, now we get to talk about the Calvin cycle. We get to take that ATP and that NADPH, and we get to use it and store it into organic compounds. So these organic compounds that we're going to be making are sugars. Usually we'll talk about glucose, but other very, very common ones are fructose is a very common one. Sucrose for plants is a very common one. It's a simple sugar. Very, very common to see. All right, so the Calvin cycle is a series of enzyme-assisted chemical reactions, and you're going to have an assignment on it to kind of help you go through it. But what the Calvin cycle does is it creates a 3-carbon sugar here. Excuse me, not here. It creates a 3-carbon sugar here, bloop, and here. And this one here, this G3P, will be used to make glucose. So what we're doing is we're taking carbon dioxide, 3 carbon dioxide from the, oh, this got cut off, sorry about that, 3 carbon dioxide from the atmosphere, and we're creating half of a glucose molecule for each run through the Calvin cycle. All right, so carbon fixation is the incorporation of carbon into organic molecules. And in this process, we're going to take 3 total carbon dioxide molecules entering the Calvin cycle through the stroma of the chloroplast, through that liquid. And then we're going to get this kind of reaction going on. In your chapter 6 packet, there's a walkthrough for this. It makes it a lot easier than me explaining it to you because there's a lot of things going on. This is your first kind of biochemical thing we're going to do. Do that before we talk about alternative pathways. So pause here, go do that, and then come back. All right, now that we've talked about the C3 the regular method, let's talk about alternative methods. Plants that evolved in hot or drier climates fix their carbon through alternative pathways, and they're going to use these little stomata on the back here, and there's little kind of crescent moon shaped valves kind of looking things, but they are small pores that are located on the underside of the leaves. The reason that we have these is that plants can control how much water is coming in or out of them. Plants normally lose water, and if these pores are open when it's hot and dry, water will escape. So why do plants need stomata? And this is so that they can bring in carbon dioxide and release oxygen. More carbon dioxide means they can make more sugar. Getting rid of oxygen is a byproduct of their waste. They need to maintain homeostasis. So if a plant accumulates too much oxygen or doesn't have enough carbon dioxide, the photosynthesis is slowed. So to combat this, plants have evolved these alternative pathways to store carbon, which they can use at night in the Calvin cycle. So they still run the light reactions during the day, but then they use the Calvin cycle at night. The C4 pathway. This allows plants to fix carbon into 4 carbon compounds instead of that 3 carbon compounds we talked about. We call these plants C4 plants. And during the hot day, they close their stomata partially. They kind of pinch it off to prevent the loss of water. Even when their carbon dioxide molecules are low, they can still fix carbon into molecules. And this extra carbon is then used to run the Calvin cycle throughout the night. Examples of this include corn, sugarcane, crabgrass. And compared to C3 plants, they lose half as much water during the day. So they're much better adapted at surviving hotter climates. Now, even more adapted are plants that have the CAM pathway. And CAM is an abbreviation for crassulacean acid metabolism. And this is named after the group of plants, or the jade plant, where this pathway was discovered first. Other examples of plants that are really hardy that use this method include cactuses and pineapples. These guys are going to open their stomata at night and close them completely during the day. And because of this awesome adaptation, they're able to require less water than either the C4 or the C3 plants. The CAM plants are the ones that are the best at maintaining water. And this is why you can see cactuses in really, really hot environments where you wouldn't see other plants. All right, a real quick summary of photosynthesis. The light reactions is where we're taking that energy, absorbing it from sunlight, converting it to chemical energy, and storing it in the form of ATP and NADPH. The Calvin cycle, we are taking that carbon dioxide and energy from ATP and NADPH and converting them into organic compounds. So light reactions have to happen during the day. The Calvin cycle can happen at night. There are some factors that affect photosynthesis, and they are light intensity, carbon dioxide levels, and temperature. And as a rule of thumb, if the light intensity goes up, there's more light, there will be more photosynthesis. This is why in the spring, when there's more light, the plants start to unfurl their leaves again and start to photosynthesize again. They have enough light to do this process. If the carbon dioxide levels go up increased level of carbon dioxide, it stimulates photosynthesis. More carbon dioxide, more photosynthesis. And the third thing is temperature. An increase in temperature increases photosynthesis until a certain point. So looking at these charts here, for light intensity, as it goes up, light intensity goes up, the plants go up to a certain point, but they go up. For carbon dioxide, if you increase it, it goes up. They flatten out, but they keep going up. For temperature, it goes way, way up as the temperature goes up, and then as it gets too hot, you start to lose too much water, and it goes way, way down. So light intensity in carbon dioxide, big increase. Temperature increases to a point. This is the conclusion to Chapter 6. I hope you guys enjoyed it. This is kind of your first step into the biochemical world of biology. We're going to talk about cellular respiration next, which is basically the opposite of this. We're going to talk about how we take those glucoses from plants and break them down into smaller substances. If you have any questions, get a hold of me. I hope you enjoyed the video. I'll see you in the next one.